So, when last we left off on DNA replication, we had been discussing helicase, topoisomerase, and SSBPs in a phase I like to call unzipping and unwinding. And that means the next phase is getting going for replication. So, in unzipping and unwinding, we learned about topoisomerase doing its job here to prevent supercoiling, helicase doing its job to unzip things, and then in eukaryotes, SSBPs preventing our strands from rebinding together. Now, let's talk about a phase we'll call priming the replication engine. All right, so what does that mean? That means that now that we've separated our strands here in unzipping and unwinding, we can't just immediately start adding new DNA to each template strand in order to simply just replicate the DNA. Why? Basically evolutionary baggage, right? If it ain't broke, don't fix it applies in biology because we always go for the laziest possible solution. And if the first original solution works, don't make something more complicated. And for some reason, the first original solution requires what we call a primer. So, in order to actually begin replication, we require an RNA primer to be added to our DNA templates. All right. And this, of course, is going to require one of the 6 enzymes involved in replication, right? We've already talked about helicase and topoisomerase. Those are 2 of the 6 enzymes. Then SSBPs, which aren't an enzyme, but are an important structure for replication. So, this enzyme that puts down an RNA primer is called primase. Who knew that primase adds primers? Now, these are around 5 to 10 base pairs long. The length isn't important for getting any answers correct, but they are short primers. And basically, new DNA, the enzymes that will add our new DNA, have to be attached to primers. So those enzymes can only start adding DNA when they are attached to a primer. So primase is over here in what we like to refer to as the replication fork. And its whole job being just put down primers. Now, what does a primer look like? It's pretty simple. In our drawing, it's just going to be a line, a little line that starts at the origin. So, we'll use green. Now, what is the origin of replication? It's where replication started. Since the easiest illustration bit is to illustrate at one of the terminal ends in our linear DNA, this is our origin here. There are DNA, like chromosomes are extremely long, so there will actually be multiple replication complexes within these chromosomes working together in order to actually replicate DNA. But so origins can be in different places. For our purposes, since we're illustrating at the terminal end, our origin is the terminal ends. So here's primase, and it's just going to lay down a primer. So Not too bad. All right. It's important to know these RNA primers are complementary to the template. Right? So if our template has A, T, C, C, T, G on it, then our primer is going to read U, A, G, G, A, C, right? Because it's complementary. A to T, G to C. But you'll notice we put U on our primer here because RNA doesn't have thymine, it has uracil. So A to U, G to C for these primers. Just a reminder that all of this is complementary base pairs. The other thing to remind you of is that we have a leading and a lagging strand, right? The leading strand and the lagging strand. And these are the strands with the two different directions, right? One with a 3' carbon on the terminal end, the other with a 5' carbon on the terminal end. And our drawing here is just going to show these primers put down. There we go. And there we go. And we're just going to remind you that RNA primers, being complementary, also run in the opposite direction. So the leading strand is 3 to 5. The primer is 5 to 3. And then here on the lagging strand, where it's 5 to 3, the primer is 3 to 5. So this will be important. Now, it is also important to note that adding primers works a little bit different on the leading strand versus the lagging strand, right? So on the leading strand, one primer. One primer at the origin, one and done, right? That's it. So, primase, adding a primer, pretty simple on the leading strand. On the lagging strand, things are a little different, right? So, on the lagging strand, primase is going to add multiple primers. So we're going to have multiple primers on here. So that's just worth noting. And this is how Primase do. This is the entirety of our section called Priming the Replication Engine. Primase adds primers, one and done on the leading strand, multiple on the lagging strand. And we will come back to talk about what happens when we actually get down to replicating.